Good afternoon. I'm Jeff Allen, President and CEO of Friends of Cancer Research. Thanks very much for joining us for part 2 in our series on the future use of complex biomarkers. Last week, we had the opportunity to hear from several leaders involved in the discovery, development, regulation, and application of biomarkers, as well as an introductory overview of the TMB harmonization project that we've been fortunate to lead over the past several months. Today, we're very pleased to be able to present the latest data and results from the TMB harmonization project. I first want to thank all of the collaborators that have participated in this work. In particular, the 17 different laboratories and test developers that came together to design this project and share their data in order to better understand how different tests used to measure TMB relate to one another. Before we get to the results discussion, I want to introduce and add great thanks to the chair and founder of Friends of Cancer Research, Ellen Siegel. Ellen?
Good afternoon. It's appropriate that the introductory music was of all these 4 seasons, and I guess we're all hoping for better seasons. But in the meantime, this is the second virtual meeting that Friends has held. A great and a giant thank you to all of you for your hard work and your partners and the committed Friends staff and science team for your work. Ultimately, this is going to make a huge difference to patients, and we are thrilled to share new data from you. It is my honor to introduce you to Dr. Ned Sharpless. Now, I have a long introduction for him, but I guess at this point, if you don't know who he is, you probably shouldn't be on this call. But let me say this, we are thankful for having him, we are dedicated to him, and we are extremely lucky to have him at the helm of the National Cancer Institute now. He is doing 10 jobs at the same time, but all of them are going to help cancer patients and help all of us, and we are deeply grateful. And as one who speaks from the patient community, a triple thank you to you, Ned, because I know your work will make a huge difference to all patients, and we're very appreciative and lucky to have you, and thank you. And with that, we'll get started with what we have to say. But again, Ned, just great appreciation for all of your hard work and everything you're doing now on cancer and COVID and everything else. Thanks.
Great. Thank you for the introduction, Ellen. It's really nice to be here. I think Friends of Cancer Research is aptly named because it really are our friends in many ways, and it's nice to see some of you at least virtually. It's sad to be not able to do these things in person, but I think it's also a little bit inspiring or significantly inspiring how much we can get done in this virtual format. The NCI I think has really risen to the challenge of disbursing grants and providing funding and catalyzing science, even via telework, and forum like this or fora like this prove that we really can make a progress for our patients, which is still sorely needed. I will be brief because I think the scientific presentations on a topic I consider very interesting will be really what we want to get to. The topic today is tumor mutational burden, TMB. I think everyone probably knows a lot about that. I will remind everyone, relatively new finding. I can really remember within my scientific lifetime, the understanding that TMB and mutational burden of cancer was really a predictor of response to immunotherapy. That is still something we just recently learned. And we think we understand why. It has to do with the presentation of these neoantigens on tumors that are recognized by cellular immunity. And so we think it's gone from sort of observation to clinically useful finding in really record time when you consider how recently it was we started analyzing the TCGA data for response markers for immunotherapy. It's clearly clinically significant now in specific tumor types like non-small cell lung cancer, and has led to this very exciting recent approval of pembrolizumab in TMB high patients based on the assay from Foundation Medicine as used in Keynote 158. Could I have my slide, please? I have one slide, which is to show how excited I was about this, that I actually turned to Twitter, wearing my mask, because we all socially distance on Twitter, to talk about what I think, why this is such an important development, because it provides a new therapy for patients. And particularly, you really have to get some sequencing to identify TMB. So I think it's a further argument why we need to have a better molecular characterization of all patients with advanced cancer for whom there is no standard of care. I'd like to congratulate the FDA. This is, I believe, tissue agnostic approval number 4 for the FDA, which is, is really almost an unthinkable paradigm in cancer, but for a few years ago. And the FDA is very aggressively moved in this direction, which I think is really great for patients. And I think is really affects the vision of Rick Pazder and others who work in the Oncology Center of Excellence. As I like to say, it's the most avant-garde part of the FDA with regard to testing the limits of regulatory, decision-making for patients. And I think this is clearly an example of how patients are benefiting from this sort of nimble thinking there. So, this is all great progress, but there's still a bit more work to do. We have to understand what TMB means in the context of other biomarkers, and the NCI is funding a number of important initiatives in that regard. And we also need to make sure this assay is analytically validated and harmonized and works across different patient populations and different labs. And that's really the work of the TMB harmonization project that has been so ably led by Friends of Cancer Research with numerous academic collaborators and at least 2 government agencies. And I think it's a really important set of developments for patients. Harmonization and standardization is much harder than it sounds. I think we have all know that, and therefore this sort of collaboration has been a really great partnership, very important in the NCI, and so it's been great to be a part of this. So thank you for the opportunity to kick things off, and I really look forward to hearing some of the data myself. Thank you.
Great, thanks very much, Dr. Sharpless, for your comments, and to the entire NCI team that has truly been a foundational part of this TMB harmonization project. One quick question for you before we let you off the hot seat today. What do you see as the future direction of these types of multi-stakeholder collaborations, and in particular, the role of the NCI research in this area around biomarker validation?
Right, well, I think the, one thing we've clearly learned from this is that TMB is quite useful, but, and even all the technical aspects notwithstanding, which need some work, but it's still not good enough by itself. So really the questions become, how can you use additional biomarkers or a better understanding of biomarkers to really predict who's going to respond to certain therapies like immunotherapy? But, certainly we have this interest in non-immunologic therapies for cancer as well. And so one initiative we have in this area is this PACT initiative, which is a partnership, a public-private partnership assembled by the FNIH to join pharmaceutical companies, which are supplying expertise and materials, along with some funding from the Cancer Moonshot and other NCI sources, to really try and look at the full complement of biomarkers in patients with various kinds of cancer, to see if we can really identify things that will go beyond TMB to predict response, so that we can really efficiently identify patients who are going to benefit and efficiently find patients who should get some other therapy. So, the understanding of response is a key question for the NCI across all domains, in an area where we've really, I think, got some exciting work going on for immunology in particular.
Great. Well, thank you for joining us today, and thank you for all that you're doing. I'd like to turn now to our 2 results presentations for today's meeting. First, we'll hear from Dr. Mickey Williams, the director of the Molecular Characterization Lab at the Frederick National Lab, followed by Dr. Lisa McShane, the chief of the Biometric Research Program at the National Cancer Institute. So first, we'll turn things to Mickey.
Present this work today on behalf of the TMB Harmonization Working Group. And I want to thank Jeff, Diana, and the Friends of Cancer Research for sponsoring this effort. It's a very important effort and yielding great results, I do believe. Briefly, as a way of introduction, tumor mutational burden, or TMB, refers to the number of somatic mutations in the defined region of a tumor genome. It's commonly reported as mutations per megabase. TMB is a predictive biomarker and has been shown to correlate with clinical benefit from cancer immunotherapies. So a very timely topic that we're discussing today. The current gold standard for measuring TMB is whole exome sequencing, or WES. Targeted panels, which assess a reduced region of the exome, have been shown to be able to estimate TMB, thus providing a more cost and time-effective way to quantify TMB in the clinic. Since panel-derived TMB scores are estimated and reported differently, it is imperative to ensure that panel TMB values are reliable and consistent to help predict patients who would benefit from immune checkpoint inhibitors. So Friends of Cancer Research convened a multi-stakeholder working group to conduct studies with the goal to generate evidence to drive alignment or harmonization, as we call it, of panels and develop consensus solutions. The methods that were applied in this project are overviewed in this particular slide. We examined 3 different tissue sources, as shown on the left. The TCGA validation cohort, which was 4,065 tissue samples representing 32 different cancer types. We did a wet lab exercise where the MOCHA lab ran whole exome sequencing, and each of the laboratories ran their own next-gen sequencing TMB panels. And this was done with 10 matched human cell lines, cancer cell lines, and matched normal representing lung and breast cancer. And then finally, we just completed the sequencing of FFPE clinical samples, starting with the N of 35 samples representing lung, bladder, gastric, and colon cancers. In the middle here, we used and defined the gold standard to be whole exome sequencing, and we agreed upon a TMB algorithm that would be used for whole exome sequencing, and it has been applied. This algorithm can be found in the first publication from this group. All panel data was compared to this whole exome scoring, and as I will show, it became apparent that the variance does exist between panels and whole exome sequencing. And there was an effort then to devise a calibration approach to better align the estimates coming from panel sequencing. And I would like to point out that Lisa McShane will follow and give more details on the calibration data that's been generated. This next slide has been previously shown, at least the left panel and the middle panel, and it describes the variability and estimated associations between panel TMB and whole exome sequencing, whole exome on the x-axis and the panel on the y-axis. And you can see that there's similar variance across the different panels here in the different tissue sample sets. And the take-home message I'd like to make is the variance is less, around 10 mutations per megabase and less, whereas the variance increases in the higher mutation per megabase base. Certainly up around 40 mutations per megabase is where the variance increases. As expected, you can see in this next slide that there is a dependency on cancer type in the actual panel NGS TMB scoring. An interesting observation from this TCA data is that the lung adenocarcinoma and lung squamous cell cancer, as well as head and neck, had less variance throughout the span of the TMB estimations. And that is compared to bladder, endometrial, as well as colon cancers, which show a greater variance. In the next slide, you see the basic calibration approach that has been applied. This is a brief schema of what Lisa is going to discuss. So on the left, 2 reference tissue sources were utilized to generate the calibration methods and curves. And then those methods were applied to the clinical specimens shown on the right panel for a form of validation. And again, stay tuned for Lisa's discussion. So in brief, a summary of our findings at this stage are our large collaborative study. We were able to describe the variability and association between many different TMB panels as compared to the gold standard whole exome sequencing. And we identified that this variation was more or less similar in its aspects, with the smaller mutations per megabase TMB scoring having less variance, and that variance increases at the higher scores. The TCGA data represents over 4,000 samples in 32 different cancer tissues and has a very nice distribution of TMB scores. But this was all in silico data. So we do believe the cell lines do complement the in silico because they do pick up the wet lab, as that all of the participating laboratories sequence these cell lines, and the data was compared across that too. And the ongoing work will continue to focus on these calibration curves and methods, and Lisa will get into that. But again, on this slide, we see that some of the take-homes were the actual panel size and its impact on sensitivity and specificity. We don't have time to get into that data, but it will be presented in an upcoming manuscript. And briefly, just to say that panel size, obviously, as far as linear sequence and the cut point used for calling TMB will definitely impact the overall specificity and sensitivity. And basically a panel size of around 0.6 to 0.7 megabases of sequence or larger seems to give acceptable sensitivity and specificity. And again, details will be provided in an upcoming manuscript. The gene content is very important too. And there was a good examination of pathogenic variants. Many panels focus on oncogenes and driver hotspot mutations as well as tumor suppressors. And by removing these common and frequent mutations, one can lower the estimated panel TMB scores and often bring them into closer alignment with the gold standard whole exome. And then germline filtering is another important topic that will be addressed in the upcoming publication. But what we know is that not everybody uses the same approaches for germline filtering. Some panels don't sequence germline tissue and rely on in silico or database removal and filtering of germline. And what we can say at this point is it seems that just using a database approach is not going to be sufficient to get alignment with whole exome. So we need to also understand the impact of poor quality samples. As I mentioned, we started with 35 clinical samples, but only got 29 sequenced because 6 were of such poor quality that they just did not report accurately, and they were flagged, and we need to continue to examine that. There's been a working group started about FFPE DNA damage, and we'll hope to be able to report out results in the very near future. So in conclusion, things that this effort has done is it was convened almost 2 years ago, and it was early in the development of this complex diagnostic of TMB. And that helped identify key challenges and helped streamline the measures to solve them. This is truly a collaborative approach, and that was needed to improve the consistency and reliability across all of the panels that have been participating, and help move the clinic as far as positively impacting clinical validity and clinical utility as this marker continues to mature. And then finally, scientific and regulatory approaches were definitely needed for alignment on the optimal thresholds and standards that were utilized as part of this ongoing effort. So with that, I'd like to thank the many people that made this possible. Government agencies listed at the top. We had great academic collaborators that were involved, diagnostic companies, as well as pharmaceutical industry, all engaged in this. And a special shout out to Precision FDA and SaraCare, who provided a lot of support. And I'll finish up there and turn it over to Lisa, and thank you for your time and interest in this topic. Thank you.
I'm pleased to present to you today additional results arising from the Friends of Cancer Research TMB Harmonization Project. Thanks to Mickey Williams for providing an excellent overview of the entire project and our progress to date. As Mickey described, the consortium has taken a stepwise approach to addressing issues associated with variation in gene panel-based assays, each with their own method of calculating TMB. We first assessed the variation that was present. Then we estimated the average relationship between each panel assay's TMB and an agreed-upon whole exome sequencing-based method. We are continuing to gain insights into factors that might contribute to differences. What I will focus on now is the development of a calibration tool to allow alignment across panels. Recall from Mickey's talk that we observed variation in the estimated association between panel and West-based TMB across laboratories. This is evidenced by the spread of the regression lines shown here. As Mickey explained, each of these data or sample types has advantages and disadvantages in terms of how it reflects the full assay process in an individual laboratory. Notice that the regression curves for cell lines and clinical samples exhibit a greater spread. While this might partially reflect differences contributed by the wet lab steps of each assay, it might also simply reflect the fact that the lines in these last two figures are estimated from far fewer data values compared to the first two. In other words, these lines are estimated with much less precision. Now I will provide a brief explanation of the concept of calibration. The basic idea is that if we have information about what range of panel TMB values tend to result from specimens having a known West TMB value, which is serving as the reference standard here, then we should be able to put things in reverse to obtain an estimate of the unknown West TMB value based on an observed panel TMB value. Looking at the figure on the right, which has West TMB on its x-axis and panel TMB on its y-axis, there are a few key observations. One aspect is the shape of fitted regression curve. In this case, a straight line provides a good fit for the average relationship on the range of interest, but we see a noticeable scatter of points around that line. The degree of scatter is reflected in the bounds surrounding the regression line, which are 95% prediction limits. For each fixed West TMB value, these limits quantify the range within which we expect about 95% of the observed panel TMB values to fall. We also see that the amount of variability increases with the West TMB value. This variance-mean relation had to be accounted for in the statistical methods used to fit the regression line. A substantial part of this scatter is likely due to biologic chance in combination with the selection of genes covered in the panel assay. It is important to quantify this uncertainty when we provide calibrated estimates. To operationalize the calibration process, we locate an observed TMB value on the y-axis, call this y0, and draw a horizontal line to see where it intersects the regression line and the prediction limits. Our calibration estimate for the West TMB is where the vertical yellow line dropped from the point of intersection with the regression line intersects the x-axis, call this x0. Similarly, we drop vertical lines from where the horizontal line intersects the 95% prediction limits, which are indicated by the purple arrows. And this gives us an interval of uncertainty to accompany the calibration estimate. For the example illustrated, we see that an observed panel TMB value of 15 would be translated to a calibration estimate of about 13 with an interval of uncertainty ranging from about 7 to 20. You might be thinking, how am I going to perform these calculations? Fortunately, we have built a software tool that will implement this process for you. It is a package coded in the computing language R, which is freely available and widely used in genomics and bioinformatics. We will be making this R package available, including the source code for those who want to use or adapt it for their own needs. If you would like to perform calibration for your laboratory's gene panel assay, you need to do the following. You generate a set of paired measurements, West TMB reference values coupled with TMB values measured by your assay. For example, you could do this in silico using the TCGA data that we used in phase 1 of our project. This set of paired measurements serves as the training data, as illustrated in the left in the blue box. One function in the software package fits the regression model with prediction limits and produces a figure like the one shown in the center of this slide. Then, using that calibration model, you can apply another function that will accept as input any new panel TMB values you want to give it. It will generate the calibrated West TMB estimates and associated intervals of uncertainty using the model it fit on your training data. The table on the far right shows some actual output from applying this software to data from one of the participating laboratories. For example, if we input a panel TMB value of 10.2, the calibrated estimate is 9.9 with an interval of uncertainty of 5.9 to 16.5. Note that for this particular gene panel assay, there was not a lot of bias on average in the panel TMB values relative to West TMB value, although it is interesting to note the width of the interval of uncertainty. Soon I will show you how the calibration tool worked on a set of real clinical tumor samples. Those clinical samples were not used in building the calibration model because that would be cheating. On this slide, I'm simply reminding you that we had different training sets that we could use to construct the calibration model. You can see that the calibration model based on cell lines has very wide prediction limits, and we expect that its calibration will be imprecise due to the limited number of cell lines available for model building. I've also added here a model that was built using TCGA data only from lung tumors. Recall that Mickey mentioned that we had some evidence in phase 1 of this project that the West versus panel TMB relationship might differ by tumor type. This figure illustrates the performance of these different calibration models on 10 lung clinical samples. Each plotting symbol corresponds to the panel TMB value obtained by a particular laboratory on a tumor sample. Each box plot shows the spread of the TMB values across laboratories. Some tumors have quite a large spread. The first box plot in each set of 5, the one shown in black, shows the TMB values without calibration. The second, in green, uses calibration based on all TCGA data. The third, in orange, uses data only from 8 tumor types in TCGA that showed a wider range of TMB values. We refer to this as our stratum 1 tumors. And the fourth, in magenta, uses only TCGA lung data. And the fifth, in blue, uses the cell line data. Optimal calibration would result in a short or compressed box plot with median, shown by the colored horizontal bar, near the West TMB value, which is shown by the thin black horizontal lines. We observed the following. Cell line calibration did not work well because it tended to increase the spread of TMB values. This is not surprising and does not necessarily reflect a problem using cell lines per se. We were not able to obtain material from a sufficient number of different cell lines to give this a fair chance. Given their renewable nature, their potential is worth further investigation. We see that TCGA-based calibrations often shrink the box plot, a good thing. Among the TCGA-based calibration methods, the one tailored to lung tumors demonstrated median values closest to the West TMB in 5 out of 10 tumors. We observed similar favorable performance of the TCGA-based calibration methods for the gastric samples. Medians were similar distance or closer to West TMB than for uncalibrated values for all cases except tumor number 39. For bladder, TCGA-based calibration methods yielded medians that were closer to West TMB for 6 out of 10 tumors, or about the same distance as the uncalibrated values in the remainder. Our results have established proof of principle for a calibration approach showing promise for alignment of TMB across gene panel assays. These are our suggestions for potential applications of the calibration tool. Calibration may facilitate combining panel TMB data across studies that aim to evaluate correlation of TMB with tumor response or survival outcomes and determine clinical cut points. Calibration may be useful for individual laboratories in their assay versioning efforts. As these TMB panel assays are rolled out for clinical use, and some are already there, calibration can assist in tailoring TMB values or cut points for clinical decision-making to the specific gene panel assay, much like normal range for some laboratory tests is assay or laboratory specific. Quantification of imprecision in panel TMB values relative to West TMB could also be useful, especially when values are near clinical cut points and when other factors may be brought to bear in making a decision about treatment with immunotherapy. I would like to thank everyone involved in the TMB Harmonization Consortium for their critically important work and for making today's presentation possible. I also want to offer special thanks to the following individuals for their extraordinary efforts preparing the results presented today amid the many challenges presented by the current pandemic situation. And here again are the members of the full consortium. Thank you for your attention and interest in our work.
Great, thank you so much. Both Mickey and Lisa will be available for questions following our next discussion. And in order to submit questions to them or any of our panelists, please use the Q&A function at the bottom of your toolbar. We have a great lineup of experts to discuss these findings, and I'd like to turn things over to our moderator for today's discussion, Dr. Victor Weigman, Director of Translational Genomics at Q2 Solutions. Vic?
Thanks, Jeff. Yeah, I'm really proud to present some of the actual friends for everyone to hear some of their insights on this importance of this biomarker, and also to ask questions that are burning for each of us as we kind of identify how best to leverage it. So yeah, I'll start off. Please continue to ask questions. We're going to hold all of them towards the end. So questions you have for Ned or Lisa or Mickey in addition, please continue to use the Q&A function and we'll fire those up. But I'll go ahead and get us started because I know with a crowd this large, we're going to have a lot of questions. I'll start off with Nicholas Botwood, VP of Oncology Clinical Development at Bristol Myers Squibb. If you care to take a few comments or introduction.
Thank you, Victor. Thank you for the introduction, and thank you to Friends for this wonderful collaboration and the tremendous progress that's been presented today. It's been a great experience collaborating with Friends and the other collaborators, so thank you for that. I lead the clinical development for oncology at BMS. We've been very invested in tumor mutational burden really since the early days in melanoma, and then with a key focus on lung cancer, and we're continuing to explore it. There's a lot still to be done, and we're excited about the discussion today and the part one discussion that was last week. So thank you for this opportunity. Victor.
Thanks, Nicholas. And next we'll bring up Dave Fabrizio, VP of Translational Strategy at Foundation Medicine.
Thanks, Vic. And a special thanks to Ellen, Jeff, Mark, and Diana, and the rest of the Friends team. This is such an important and vital effort. And, I've had the privilege to be a part of it, like many of my colleagues, since the beginning for the last several years. So, truly we have become friends over the course of that time. And so, I'm excited about the results that Lisa and Mickey presented today, and looking forward to a good discussion. I lead translational strategy at Foundation Medicine, where we developed Foundation 1 CDX, the assay that was recently approved for TMB high patients with pembrolizumab across late stage solid tumors. So great to be a part of the discussion today. Thanks again.
Thanks, Dave. Following up next is Reena Philip, Director of Division of Molecular Genetics and Pathology at the Office of In Vitro Diagnostics and Radiologic Health inside the CDRH at the FDA. Reena?
Thank you. Thank you, Friends, for this great project. Diana, Jeff, Ellen, it has been a really great project, and happy to be part of this project from the very beginning of this. So at FDA, I'm at the Office of In Vitro Diagnostics, Division of Molecular Genetics, where we regulate the molecular in vitro diagnostics devices in oncology. Thanks again.
Thanks, Reena. And last on our panelist is Naiyer Rizvi, Director of Thoracic Oncology and Co-Director of Cancer Immunotherapy at Columbia.
Everyone, thanks for inviting me. It's exciting to be here. I think back to when we first published data around TMB as a predictor of response to immunotherapy 5 years ago, and the tremendous progress that's been made. It's been really remarkable, and I'm happy to be part of this effort.
Thanks. And yeah, Naiyer, since you're last, we'll go ahead and queue you up with a question here. Ned talked a little bit about TMB and what it's meant to convey, but it is a complex biomarker. That's why we're all here to talk about that. And you were one of the earliest people to kind of bring that to light in lung cancer. And so can you talk a little bit more about what makes it complex? And pulling these variants from the exome as a surrogate for what's happening in the tumor genome has a biological impact. Could you elaborate a little bit on that? And before I mention that to all of our panelists, I'll allow about a minute and a half for questions because we do have a lot coming through the pipe here. So you will hear a little ding at the end to make sure that we give everybody enough time to answer the questions we have before us. Naiyer?
Right. So, I think as has been mentioned, TMB really denotes the number of acquired non-synonymous somatic mutation in the entire coding region, which is sort of 30 to 35 megabase panels that we use now for whole exome sequencing. And I think initially we weren't really sure why TMB was important, but I think what we know now is that high TMB just leads to a stochastic increase in likelihood of a small number of really true immunogenic mutations that are able to bind to MHC class 1 with high affinity and be recognized by a T cell receptor with high affinity. So when you have that occurring, and you give immune checkpoint blockade, you reinvigorate T cells against these unique neoantigens. And so if you have a high TMB, you have more neoantigens, you have a stronger response to immune checkpoint blockade. I think one of the challenges is exome sequencing is more challenging, time-consuming, requires germline. You might not be able to assess driver oncogenes of interest as well. So there's been a huge move for these larger focused NGS panels, but they're in generally about a megabase in size rather than 30 to 35 megabases. So therein lies the challenge.
Thank you. Yeah, and so as we extend beyond that, how we leverage it, you know, Dave, I'll move to you to kind of talk about, you know, what impact this has in test development. So what are the findings that we're having here as friends, right? Mickey and Lisa presented some of this data that's going to be coming through in our upcoming publication. But what are the things that are going to have the most impact to the field from all of us working together to create this common understanding of the marker?
Yeah, it's a great question, Vic. You know, I think there's a few ways to answer this, but I think first and foremost, just the idea of adding a fourth aspect of harmonization to the already important analytic validation studies most of the diagnostic companies are used to performing, that include accuracy, precision, sensitivity, the ones that we outlined in the phase 1 manuscripts published just a few months ago, really provides a framework to ensure consistency of the calculation across the diagnostic platform. So not that harmonization should replace, you know, accuracy or the need to establish clinical utility, but I think it now becomes sort of a prerequisite for ensuring apples to apples comparison. I think secondly, the results that Lisa and Mickey presented today show that, you know, we're actually starting from a pretty good standpoint, meaning the variance across platforms, you know, seems to be within reasonable limits for most of the participating labs, and that's even before any calibration was actually performed. You know, I think one of the keys will be to understand, you know, whether at certain cutoffs, like 10 mutations per megabase, the variance we observe across platforms is actually within the precision of any one individual assay, meaning it may or may not be negligible. I think the last thing is that the recommendations, you know, around the factors that influence TMB that Mickey mentioned, including minimum size of a panel, the different filtering mechanisms, these can ensure that we build similar methods to reduce future variance as much as possible. So I think this has applications for, you know, diagnostic companies currently developing their TMB methods. You know, this will ensure consistent clinical application of TMB across trials, you know, also for therapeutic decision makings, but also an apples to apples comparison of retrospective studies, perhaps with different TMB platform values.
Thank you. Yeah, I agreed on all points, right? And so, you know, I'll move us a little bit forward to figure out how do we take these recommendations to change or address or adopt our current practice, and how we facilitate this work in our own fields, right? So since you talked about Nick, I'm going to go right back to him briefly to talk about, you know, diagnostic and drug development, right? We want to use the marker here to identify patients that are going to perceive and recognize the most therapeutic impact. Can you talk about what defining this marker is going to be involved in that process?
Yeah, thanks, Vic. And you know, this is tremendous progress for everybody who's involved in cancer care, and ultimately for patients. And it's just incredibly important for us to have standardization of this test so that we are able to compare across trials and across data sets, so that we can best inform research and ultimately best inform treatment decisions for patients. Because I think we've made a huge amount of progress with tumor mutational burden, and it's an important part of the framework for decision making. I don't think in the context of immunotherapy and checkpoint inhibitors, it's the whole answer. The immune response to cancer is extraordinarily complex, and there are probably many factors that we still need to understand. But having some sort of standardization of TMB, so we're comparing appropriately and we're understanding what the relevant cutoffs are and how they look across trials, is very important, not just when we conduct studies in the US, but across the world, and working with health authorities and patients all around the world. Many of these questions that remain aren't going to be possible to be answered until we're using the test consistently and we know what we're comparing against. So for example, there are many other potential markers that we need to look at that have relevance to TMB, like neoantigens, T-cell receptor clonality, for example, as other markers. The relationship with plasma TMB, we currently are focused on FFPE, I think appropriately so, but plasma TMB may be an even better predictive marker and more representative of the tumor, and we have to be consistent in that. I think ultimately, as we move to selecting patients and we think about the clinical validation and the clinical utility and building on all the fantastic work that's been done and facilitated by friends, and we move more to prospective studies, it's going to be very important to understand what the cutoffs are and how they apply to different tumor types. We know that tumors have different levels of TMB inherently, and there may be different cutoffs that have different clinical relevance. Added to which, most of our data that we've generated to date is based on PD-1 monotherapy data. There may be different applications as we move to combinations of checkpoint inhibitors and move the field forward in that way. So tremendous progress and a foundation from which I think we can really build and take the therapeutics to the next level.
Yeah, thank you. And I'm going to use your kind of segue here into clinical practice to go back to Naiyer before I have Reena kind of elaborate us what the regulatory impact of that is. You know, Naiyer, clinical practice. I mean, we have cutoffs for TMB, right? And then we're running our patients into. But can you go a little bit more about how the TMB value gets distilled into clinical practice, right? And what test selection and meaning of that marker means as you treat patients across these different therapeutic options.
Right. I mean, I think at a practical level, you know, I think that this is going to change how we prioritize or position these targeted NGS panels. A lot of places may deplete the precious amount of tissues from doing multiple IHC stains or you know, doing PCR for you know, for MSI testing, and then you may be in a position where you don't have enough material left to do your you know, your targeted NGS panel, whereas you can get a lot of that information or all of that information if you position the targeted NGS panel first. So I think that we, you know, as treating physicians, will need to work with pathology and laboratories to try to move target NGS panels earlier in the paradigm so we don't deplete tissue inappropriately.
Yeah, and of course that means you're going to be using the test that we get approval for, right? And so, you know, Reena, it naturally goes back to you, kind of representing your thoughts on, you know, as Ned called out in the beginning of our discussion, we do have a, you know, pan cancer opportunity here for using TMB. You know, can you elaborate a little bit more about what that kind of tissue agnostic approval means with high TMB from the FDA setting?
Yeah, so, you know, as you all heard from the earlier talk from Mickey and Lisa, the TMB harmonization project was looking into the different sources of variability in the TMB scores across panels. You know, we heard panel size, gene content, gene coverage, germline filtering, you know, types of exonic mutations included in the quantification, proprietary algorithms. Those are important sources of variability when you are harmonizing the definition of TMB. So moving forward, at FDA, when we compare device performance across different platforms relative to the specific clinical cutoff that was being approved, for example, in the recent tissue agnostic approval was for 10 mutations per megabase cutoff. So it is important to understand whether this TMB high determined by this FDA approved test, is it comparable to the TMB high determined by the other platforms relative to that cutoff? And, you know, we look into the qualitative results, and we also look into the underlying factors that will maybe contributing to the differences in this TMB score. So it is actually, we'll be looking to understand whether these devices compare across the range of scores relative to the cutoff. And you also heard the TMB project was, you know, in the phase 1 manuscript, it was proposing best practices for analytical validation studies. So when doing the review of this tissue agnostic PMA, we did look into those best practices, as you probably could see it from the SOCD, Summary of Safety and Effectiveness. we requested they perform the alteration level accuracy and precision. And that really helped us to understand the discordant results between the approved CDX and the whole exome sequencing. And also, as part of the TMB project, it's also trying to ensure consistency in TMB calculation through this alignment using this universal reference method. And this calibration curve generated, it could promote alignment of and analytical validation of these panel TMB values. It could be a supplement to the analytical validation.
Thank you. And, I questions for you are certainly in higher demand. Now, so I'm going to double up on you here real quick. So, someone's coming, they have a potential trial result they want to show with TMB on their test. What do they need to bring forward? You just talked about the calibration tool again, but what are you looking for these people to show for importance of that TMB with association to that drug?
Sure. So if they are looking for a follow-on companion diagnostic claim to pembrolizumab, they do have to show clinical validation, which is actually a non-inferiority study using clinical samples. It could be procured clinical samples, but it's from the intended population, so it should cover the range as the different tumor types. As you can see from the drug label, they had different tumor types, so it should cover those tumor types, and they should cover the range of scores. So those clinical samples could be used for clinical validation and show non-inferiority study to Foundation Medicine device, which is the approved device now. This calibration could be actually, like I said earlier, it'll be a supportive analytical validation, in addition to the other studies that are also in the SOCD for the Foundation Medicine FOCDX PMA.
Yeah, no, absolutely right. I mean, thank you for that. I know a lot of people have stuff in their pocket they're wanting to use. And I will say, back we talked about calibration, we talk about we're trying to standardize, all of us, and we're all from different parts of the drug development process, but we all want to be able to have our tests be meaningful. We talked earlier about a cutoff of 10, which sounds certainly arbitrary until you do, as you mentioned, that clinical utility measurement. You have to show the trial that has the utility you want. So I'm going to actually bounce back to Nicholas again, and then Dave, I'll allow you to follow up just a little bit on, what is 10 the same for everything? We saw some of Lisa's results with her plots of showing, okay, they could be relatively similar, but how do we standardize? As all friends here for the TMB working group, how do we standardize on what that value means so that the tests we're running here, that Naiyer would run at Columbia versus tests run in Florida are all measuring the same thing? How do we get to that point? How do we get to envisioning harmonizing on that role? And Nick, I'll allow you to start, and then Dave, a short follow-up from there.
Yeah, thanks, Vic. This is an important question. I don't know that 10 is necessarily standard across all tumor types and for all settings.
Probably not, right?
We have to think about frontline. We have to think about refractory disease combinations, different tumor types. We know the level of TMB is different in tumor types. We did a lot of research in collaboration with many on the panel today in lung cancer, trying to optimize the cutoff, phantom mutations per megabase to be an appropriate cutoff to enrich for responsiveness, certainly. But we must remember, of course, that TMB is a continuous variable. This has been talked about a lot, and I'm sure everybody on the call is aware of this. It's a quite different concept from a sort of dichotomous variable like a driving mutation. It is a continuous variable, and frankly, different cutoffs may be different in different settings. In terms of, again, the clinical utility and the validation, important considerations in this, a lot of the data that's been generated to date has been retrospective. One of the challenges we've had is ensuring samples on all the patients, and that the patients we have samples on are representative of the overall treated population. This is often a challenge in studies and something that certainly the FDA and other regulators really focus in on. So ideally, prospective studies are key, and we have some of those to really optimize the predictive utility of the test. And that is going to be very important. So I think these are some additional considerations in terms of how we validate the cutoff.
And Dave?
Yeah, maybe I'll add on to that. I agree with a lot of what Nick said. I think what essentially the harmonization consortium has done is provide a framework to reduce the variability as much as possible across platforms so that, 10 on F1 CDX, can be as close to 10 on the other platforms, to the best extent possible. And I think the calibration tool that Lisa described, is a real meaningful tool that can actually help new companies or companies looking to develop a TMB assay align as much as possible to F1 CDX or to whole exome sequencing to ensure consistency of that calculation. So from an analytics standpoint, I think that some of the tools that were discussed today can help ensure consistency. Regarding the continuous variable nature, I can't agree with Nick more on this point. We, at the end of the day, a binary decision, a treatment decision needs to be made, to give therapy or not. And I think that's why we've, forced these complex continuous variable biomarkers into dichotomous high or low, positive or negative. But I think at the end of the day, it doesn't mean that the information or the data that is used to make that binary decision has to be limited to dichotomous positive or negative, right? There are continuous variables. TMB tends to be probabilistic, and we need to think creatively about how we utilize all of the information. We maximize all of that information. So actually, at the end of the day, the best clinical decision can be made for a patient.
Great, thank you. Thank you very much. We'll get some of the, I do have a couple questions with regards to the technical items, and then I'll conclude with our last few minutes of looking forward. So Mickey, I'm going to come to you real quick. We have oddly enough, a lot of questions talking about, we mentioned it earlier a couple times in our dialogue here about synonymous versus non-synonymous and what impact that has on TMB. And you actually had a slide that touched on this briefly. Could you elaborate a little bit more on that impact of protein change in the TMB and what things, yes, Lisa, you were followed up, so you queued up great. What things actually do you see from looking at the data have a larger contribution to TMB?
Well, great question. I would say yes. The method that we first chose and was published, first off, did not include synonymous mutations. It only dealt with the actual missense and insertions, deletions, and the like. One of the major impacts that I think we've picked up on, obviously, the size of the panel and how much linear sequence space the panel has. Early days, we had some small panels, much lower than 0.6, and they were not so accurate in correlating with the other panels and or with whole exome. So that's a critical thing. The one thing that I think came out of this too, in the first manuscript, detailed very well, is at what point do you have confidence in calling a mutation? If you go too low in an allele frequency, you move into the noise of the assay. And so it was decided to have a 5% cut point, at least for the whole exome. And so 5% allele frequencies and greater. That's another critical thing. And the one thing we can't get away from is the different tissues and how they play a role in this. And clearly the data from TCGA indicates not only that the TMB ranges in different tissues are very different, but also how different panels report across tissues. There's certainly a bias there, and that will have a lot to do with how those panels were developed and their gene content. So they all contribute to this, and what the largest impact is, I can't tell you at this point, but certainly being aware.
That's what we're trying to find out, right?
Yeah, certainly being aware of what we've picked up on is a critical thing. And again, I'll go back to the calibrator. I think that's going to be a great method going forward, and the fact that the data is in the public domain. As Lisa pointed out, you can actually apply your own panel and see how it compares.
Yeah, and I'll actually use that to segue to Lisa before I kind of ask our last question. We talked about the calibration tool. I want to make sure as we wrap up, as people listen more right towards the end, when will people get access? When and how do people get access? And the second thing is we're showing how similar your panel is compared to all of our all of ours in the consortia, and to what we consider the gold standard, the exome. What if somebody's panel falls outside of that confidence interval? What do they do then? So kind of a little two-part question, but the panel or the calibrator, when do we see it? The not us and friends.
So the current plan is to make the calibration tool available when we publish the paper from phase 2. That will give us time to make sure we've worked out all bugs and that we've done some user testing. You know, we'll give the members of the consortium access to it before we actually put it live. You know, in terms of how people can use it and what they do if they find that they're way out of line with everybody else, you know, I think it really goes back to the whole philosophy behind this consortium is that we have to first understand what the level of variability is across these panels, and then we have to dive deeper and figure out what factors contribute to that. So I think that if a certain assay developer finds that they are quite far away from WES, which would imply that they're quite far away from many of the members of this consortium, I think they need to be concerned, and they need to think about whether there are some factors relevant to how they do their assay or their bioinformatics that might be driving that difference. You know, I think this whole thing is really a learning exercise. And, you know, statisticians often like to think of modeling as, you know, we try to model what we can explain, but then understand the magnitude of what we can't explain. So you can think of that as I can describe my association with WES, and the amount of scatter around that line will say what I can't explain. And by understanding these different sources of variation, I might be able to reduce that scatter or move that line.
Excellent. Thank you. Thank you. And last question, I would say it's by far our most popular question. Thank you all who have participated and asked a lot of these things. But I'm going to start with Naiyer and end with Reena. Liquid biopsy, right? This consortia has done an amazing job of bringing up competitors, both on the test and the drug development side. And we've all kind of agreed how important this is for tissue. But liquid biopsy, you know, Naiyer, if I could start with you very briefly, you know, how do you think liquid biopsy changes how we use TMB? And then followed up for you, Reena, is what do we need to be considering as test developers like myself and Dave, and work with our pharmaceutical partners to do these trials? What should we be considering? So Naiyer, can you take this? And this will be our last question, and I'll give it back to Jeff.
Yeah, so I can certainly give the clinical perspective, which is, you know, the ability to, you know, have sufficient tissue or to acquire it from another lab, from another state, you know, or even across the street in New York City, it can be very challenging. And so quality, quantity, availability of tumor tissue has been a challenge, and it's been pretty remarkable that, you know, that there's sufficient circulating tumor DNA and plasma in the majority of patients with advanced cancers. So I think it's a huge opportunity.
And Reena, can you give any just insights of what needs to be provided to show?
Yeah, liquid biopsy is actually also a very, you know, topic that is dear to my heart. Actually, it's you know, I think even liquid tissue, my one you know, recommendation to all the pharma is collect samples. Collect, you know, make sure you have a robust banking plan, because that's what we see the main issue with respect to these companion diagnostics validation, that the clinical trial samples, the sample attainment rate is really low. So if they can actually have that, you know, that plan in place in all the clinical trials, you know, that could be a blood TMB, could be another potential great biomarker.
Great, thank you, Reena. And with that, thank you all panelists. I'll bring it back to Jeff to close us out. Thank you all.
Hey, thanks very much, Vic. And thanks to all of our participants in today's meeting. This project has been a true collaboration across all sectors. And while the science of TMB will continue to evolve, this consortia has developed an approach that will certainly help shape the future of complex biomarkers. And in Friends of Cancer Research, we believe that we'll get further faster by working together. I want to thank the entire team at Friends, particularly Dr. Diana Marina Vega, who spearheaded this project. And thanks to all of you for joining us today. Please stay tuned for details on our additional upcoming next virtual meeting, the next of which will be presenting the initial results from the CT Monitor project. It's unrelated to TMB, but it's another unique research partnership exploring the role that ctDNA has as an early and easier tool to measure response to treatment. It takes place on August 11th, and we hope we'll see you then. Until then, take care and be safe. Thanks so much.